Good morning. I welcome everybody to have a view on my third session analysis. Uh, my name is Jeff Ochieng. Yes, I'm going to discuss on analytical challenges. In third session, we tackle on the analytical challenges, whereby the analytical challenges we tackle on the malaria and application of next generations. We get to know the data sets and the bioinformatics resources. So the data sets, how to get the data sets from the different resources. So we tackle on how we get to know how the data sets that we can get them from different sources. Also, what a data set can answer, the data set that we get from those resources. The malaria data sets, the Ebola, also the HIV. So what that data set can answer. Also, how data set can be used. The data set that we get from bioinformatics, different resources, how are we going to use it in our next generation sequencing? In SF9s, the SF9 cells is the production of biological using viral vectors and identification of viral contamination using genomics. And the availability of the FS9 genome sequencings can help identify and characterize indigenous viral sequencing for interest and aids in chromosomes mapping. We also tackle on metagenomics and viral sequencing, whereby we has three process, that is amplicon. Also we tackle on the targeted and the whole genomes, whereby you can see the targeted amplicon sequencing and the whole genome sequencing. All these are the processes that it takes. In targeted panel sequencings, it is cost-effective way to detect known and novel variants in selected set of genes. It also has a high coverage, like you will see, with 40 to 400 genes. It's require a less samples input and produce smaller amount of data than the whole genome sequencings, making analysis more manageable. And in whole exome sequencing, in all it is intermediate coverage. It is a genomic technique for sequencing of all protein coding region of gene in genomes. It consists of two steps, whereby the first step you select only the subset of DNA that encode proteins. The second step is the sequence the exomic DNA using any high through input of DNA sequencing techniques. It is an intermediate coverage. You can see these are the steps that it entails. And in whole genome sequencing, is a process of determining the complete DNA sequencing of an organism genome at a single time. It's determine it determines the complete DNA sequencing at a single time. It has a low coverage. All the genomes and the non-coding DNA are being determined in whole genome sequencings, and it is a comprehensive method for analyzing entire genomes, and also it is also not it is cost effective. So again, we went ahead to talk on the data formats for sequencing data, whereby data format for sequencing data, we have three formats: the FASTQ files, we have the FASTA files, and the GTF files. In FASTA files, it has a quality reads. The quality read contains sequencing data from the cluster that passed filter or on flow cells. The information about the quality of each positions of the reads, it is determined by the multiplexings. Also, in FASTA file, both contain sequencing reads and also has these two files has raw data. Yes, in FASTA files, it does not have any coding information. It represents either nucleotide sequencing or the amino acid sequencings. Yeah, and also it has the raw data for sequencings. So, and GTF, it is a notation of it is identical of the GFF, the gene transfer form. In GTF, it has information about the location of various genes. It's also used to hold information about the gene structure, the gene features. Mm, yes, this one is it's work differently with these two. Yeah, these ones in the FASTA-Q files is the reference genomes. Sorry, the FASTA file. The FASTA file has the reference genome. The FASTA-Q has the sequence, and the GTF has the position. It's bring the positions. So again, we also saw the DNA variants calling, whereby the DNA variants calling, it consists of all these steps that we are seeing: the pre-processing tests, the mapping alignments. We have the variants calling. We have the different variants calling, the annotation, and post-processing. In the pre-processing, this is where we build our pipeline. Yeah, this is where we build our pipeline. Then we start when we go to the mapping alignment. In mapping alignments, we map all quality scores and all reads that match the genomes, even if the repetitives difference from most other alignments. So we align short reads to the reference genome and accurately include the SNPs, the short directs. So after all the alignments, then we have to three variants calling. The three variants calling examples: the somatic mutants, the new tools for the analysis of cancer samples and can detect mutants below 5% frequency with high quality. So then after the process of three variants callings, then we have the different variants calling. In different variants calling, the nematodes of the genomic backgrounds of the mutants is different from the reference genome. The number of backgrounds variants may exceed the number of true mutants by several orders of the magnitudes. Then we go to the annotation. The annotation is a process of identification of locations of genes and all of the coding regions in our genomics, and determine what this gene does. It will determine all the genes that are going to be run, what it's going to do. Then later we end up with the post processing. Yes, so after building up of this whole process of DNA variants callings, then we also run our pipeline. So in running our pipelines, we took this data as a SRA table. So we ran mapping of the reference genomes using the SR2, whereby the output produced was mapped, there was mapping, and also ran our pipelines using the two samples. Thank you and welcome again.